DNA replication. For DNA replication, we firstly need the template, raw substrates, which are deoxynucleotide triphosphates, enzyme and proteins, which we template and assemble DNTPs. Okay, so let's firstly start with an overview using bacterial replication as an example. So we have the double-stranded DNA, which then unwinds, followed by elongation. Okay, so let's explain further the process of unwinding. For unwinding, initiator protein and DNA helicase firstly bind to the OEC of the bacterial DNA, causing a short stretch of DNA to unwind. This unwinding allows helicase and single-stranded binding proteins to attach to the single-stranded DNA. During the process of unwinding, DNA gyrase release the strain ahead of the replication fork. The role of single-stranded binding proteins is to stabilize exposed single-stranded DNA. Okay, so for the second step, which is elongation, primase is needed to synthesize primers which are needed for elongation. These primers provide a hydrosyl which allows the binding of nucleotides by the DNA polymerase III. Following the addition of these nucleotides, DNA polymerase removes primers which were added by primase to only allow synthesis of DNA molecules. Following the removal of the primers, DNA ligase comes and seals the nick with phosphodiester bond between the 5-phosphate P group of the initial nucleotide added by DNA polymerase III and the 3-OH group of the final nucleotide added by DNA polymerase I. We then have a double-stranded DNA molecule.